Welcome to all in virtual classroom of biology for standard 12 science students produced by Department of Biology is a part of Janta Shikshan Sanstha's Kisanvi Mahavidyalaya Vai in session number 7 presented by Sri Sushant Kumar Swami. Let's see. Today we are going to discuss concept embryonic development. Embryonic development is also known as post-fertilization changes. These changes occurs in zygote, that is called as embryonic development. This embryonic development takes place in uterus. Embryonic development takes place in uterus. In sophisticated word, embryonic development means it is a biological process that forms a single cell to in a multicellular structure, that is baby. It means that single cell structure is converted into multicellular structure is known as post-fertilization changes or embryological development. Study of embryological development is known as embryology. Study of embryological development is known as embryology. Now let's see embryonic development takes place in human being. In human, embryonic development is completed in 3 steps. In human, embryonic development is completed in 3 steps. Look at the screen. These steps are cleavage. First is the cleavage. This one is the cleavage. What is cleavage? Cleavage is nothing but it is a rapid cell division. It also takes place in 3 parts, which are as follows. Cleavage is a rapid cell division. It is a mitotic cell division. It takes place in 3 steps. First one, first cleavage. It is a longitudinal. It is a longitudinal or vertical. It forms 2 cells. These cells are now known as blastomeres. This is called as blastomeres, and it takes after 30 hours of fertilization. This is the first cleavage. Before going to second cleavage, there is one important thing should be noted here. Human embryological development is holoblastic. It means that there is no specific plane of division takes place. It may be sometimes vertical, it may be sometimes longitudinal, it may be sometimes radial, it may be meridional. Horizontal, longitudinal, meridional, radial, any plane maybe takes place. You used at this division. That's why it is a holoblastic. So in first cleavage there is vertical, and we got 2 blastomeres, means 2 daughter cells, that is called as blastomeres. These blastomeres, 2 blastomeres again divides. They divide vertically but right angle to previous ones. They divides vertically but right angle to previous is known as, it got 4 blastomeres, and this stage is known as second cleavage. It takes 60 hours after fertilization. It completes within 60 hours after fertilization. Then these 4 blastomeres are again divided into 8 blastomeres. Again divides into form 8 blastomere. This is the third cleavage. This cleavage is horizontal and right angled to first once, and it completes in 72 hours after fertilization. Now this 8 cell structure is again divided rapidly. It forms 16 cell structure. This 16 cell structure is known as morula. This 16 cell structure is known as morula. This morula is again divided and form 32 cell structure. This structure is morula. Up to till 14th day are covered after fertilization. This morula ball consists two types of cells. Inner cells are larger and outer cells are smaller. Inner cells are larger and outer cells are smaller. This is a morula. Now this morula is enter or reach up to uterus. It means that first cleavage, second cleavage, third cleavage takes place in fallopian tube. Now this morula enters into uterus. It absorbs uterine fluid, which is nutritious fluid, and these cells become flat. These cells, outer cells, are known as trophoblast. These outer cells are known as trophoblast. This trophoblast again absorb more nutritive fluid, and these all cells are scattered, separate and scattered. They form a cavity. That cavity is known as blastocele. And remaining the structure all cells to form a cyst-like structure, and this cyst is called as blastocyst. Means morula cells increases size, form a cyst, and this cyst is called as blastocyst. Now this blastocyst is ready for implantation. Implantation means attachment of embryo to deep inside wall of uterus. Attachment of embryo deep into wall of uterus. Before going to that, we can see significance of implantation. Implantation gives nourishment to developing embryo, and secondly, it gives protection from jerk and shocks to embryo because it is attached in wall of uterus. This blastula, I come again back to blastula. This blastula is formed. It forms cavity. That cavity is known as blastocele. You know very well. Later, all cells are separated except one side. All cells separated except one side. The trophoblast cell in contact with embryonal knob are called as cells of Raubers. The tropoblastic cells which come in contact with the embryonal knob is called as cells of Rauber cells. And these, at this stage, blastocyst shows polarity. Important point is here. Till then I cannot say the pole 1, pole 2, but here is the polarity. Inner cell mass is embryonal end and outer cell mass is embryonal end. Embryonal and embryonal, like these poles, are observed in. This is called as polarity. Then blastula goes to implantation, and implantation we'll see previously. Let's revise it. It is a attachment of embryo deep inside wall of uterus is known as implantation. After this implantation, before going to implant, there is another point should be noted here, that a zona pellucida is a layer present outside the ovum. This is intact till this blastula is completed. It is intact. If zona pellucida is not break, pregnancy is not there, implantation is not there, and abortion takes place. For that, before implantation, zona pellucida must be degenerate or ruptured. And zona pellucida is ruptured, then and then only this blastula or blastocyst is embryo is implanted into the wall of uterus. Next procedure, this structure process go to next, gastrulation. This is a milestone. Cleavage is one type of milestone, blastula is one type of milestone, such as a gastrulation is third step of, it is third milestone. What is gastrulation? Gastrulation is nothing but conversion of blastula into germ layers. In another word, we can say formation of germ layers from blastocyst is also known as a gastrula. One point should be noted here, formation of germ cells. Means germ layers are formed. How this forms? Let's see. Formation of germ layers takes place in 3 steps. First one, formation of endoderm. Second, formation of ectoderm. And third one is formation of mesoderm. Here, point should be noted here. Endoderm first, second ectoderm, and third one is mesoderm. Why this is? Because initial stages, animals, primitive animals or smaller, lower animals, they are diploblastic. Later on, multicellular, higher animals, they are diploblastic. That's why first endoderm, second ectoderm, and then mesoderm. Formation of endoderm. Let's see formation of endoderm. Some cells covering the pre-surface of embryonal knob. In blastocyst, some cells covers pre-surface of embryonal knob. Then they rapidly multiply. They rapidly divide and redivide, multiply, increase their numbers, and spread out in all direction. Spread out in all direction. Spread out in all direction to forms endoderm. To forms endoderm. It will be formed endoderm. To forms endoderm. Embryonic knob becomes columnar to form embryonic disc. First endoderm is formed. Then embryonic knob becomes columnar to form embryonic disc. Form embryonic disc. Now the space between embryonic disc and trophoblast is called as embryonic cavity. Embryonic disc and trophoblast is space that is called as embryonic cavity. This embryonic cavity is filled by amniotic fluid. This amniotic cavity is called filled by amniotic fluid. This is amniotic cavity. Amniotic cavity is a space in between embryonic disc and trophoblast. Then formation of ectoderm. After the formation of endoderm, the remaining cells of embryonic disc, after formation of endoderm, remaining cells of embryonic disc is arranged in layer. Remaining cells are arranged in layer, then it is called as ectoderm. It is called as, this layer is called as ectoderm. Then formation of mesoderm. The cells are present on hind end of embryonic disc, starts rapidly, divides and redivides, and they spread between the trophoblast and endoderm. They spreads between trophoblast and endoderm. Spread between trophoblast and endoderm, that is called as mesoderm. This is the form of, this forms mesoderm. This which separates yolk sac and amniotic cavity. Yolk sac and amniotic cavity is separated by mesoderm. Completion of all steps, germ layers start to form organs and tissue. This process is called histogenesis or organogenesis. The organogenesis as follows, means how organs are developed. from which layers? This is called as organogenesis, and it is as follows. From ectoderm, it is called as fate of ectoderm. From ectoderm, epidermis of skin, hairs, nail, sweat gland, salivary gland, mammary gland, lacrimal gland, sebaceous gland are formed. From ectoderm, epidermis of skin means outer layer of skin, hairs, nails, they are the derivatives. Sweat gland, salivary gland, mammary gland, lacrimal gland, sebaceous glands, all are developed from ectoderm. Nasal cavity, enamel of teeth, internal and external ear, they are also from ectoderm. Even though foregut, midgut of alimentary canal, adrenal medulla, pituitary gland, and entire nervous system, all these organs develop from ectoderm. Then from mesoderm, all types of muscles, connective tissue, dermis of skin, circulatory system include heart, blood vessels, and lymphatic system. Then middle ear, dentine of teeth, urinary and reproductive duct, gonads and kidney, sclera and choroid of eye. All these are developed from mesoderm. And last one is endoderm. From endoderm, epithelium of midgut, gonads, glands of stomach, tongue, tonsils, lungs, trachea, bronchi, larynx. These are developed from endoderm. Not only these, but layers of urinary bladder, vagina, these are the developed from endoderm also, as well as thyroid gland, thymus gland, eustachian tube are also developed from endoderm. All these completed, after the completion of all these, embryo is formed and it is growth in uterus. It is some steps, developing steps of organs. Observe carefully, and this is the stages of embryo development in uterus. Let's see till then. Thank you. To be continued in next session.